Hello, everyone, and welcome to today's webcast, Contamination Control for Pharmaceutical Products, Real-World Moist Heat Validation Pitfalls, and Proper Verification of Sterility. I'm Tim Wright, editor of Contract Pharma, and today's webcast is being sponsored by Nelson Labs. They're a global provider of microbiological and analytical lab testing services for medtech and pharma companies. You can visit them online at nelsonlabs.com. We have three speakers on the line today, Martel Winters, Jason Pope, and Susan Schneep. Martel is the Director of Scientific Competency at Nelson Labs and a specialist microbiologist and a radiation specialist. He's been at Nelson Labs for 25 years and spent his earlier years working in the radiation sterilization group, responsible for bioburden testing and radiation validation studies. Jason is a senior scientist at Nelson Labs and is a certified quality auditor and specializes in providing consultations for clients about sterilization of reusable medical devices, endoscopes, and pharmaceuticals, and general sterilization validation process development. And lastly, Susan Schneep is a distinguished fellow at Regulatory Compliance Associates. She has over 40 years of quality assurance experience in the pharma industry. And Susan's going to be joining the discussion after Martel and Jason run through the slide deck, and she'll be moderating the Q&A session today. Today, Martel and Jason are going to address real-world observations relating to moist heat sterilization processes, discuss the science behind the observations, and present solutions to result in a successful validation outcome. We will be accepting your questions throughout the talk. You can send those in using the box at the bottom of your screen. And as I mentioned earlier, questions will be answered during a Q&A at the end of the 20-30 minute slide deck presentation. If you're experiencing any technical difficulties, you can type those into that question box as well, and someone on the back end here will help you out. So at this point, I'll turn it over to Martel.
Okay, welcome everybody. We're excited to have you here with us today to go through these interesting topics. Just a heads up, so that these ideas we're covering today, some of this information was actually sparked by, these are smaller portions of bigger topics that we're going to be covering in this seminar that's on the screen now in late October in Philadelphia, the 26th and 27th of October. And information on this seminar, this is a two-day in-person seminar, so this will be in person there in Philadelphia, and information on this is provided on the website at nelsonlabs.com in the events page. So if you want an opportunity to get more in depth with some of these details and others, that information is on the website. And Jason, I'll turn it over to you now.
Okay, thank you, Martel. So when we start to talk about contamination control in a manufacturing setting, one of the important things that we need to talk about, one of the important things we need to consider is sterilization and proper sterilization validation. So one of the things I wanted to do was to run through a few documents that can be used for guidance to understand requirements and understand the science related to sterilization validation, specifically to moist heat sterilization validation. So one upcoming change that I'd like folks to be aware of is that the ISO moist heat sterilization process document, ISO 17665, is actually going through the revision process. And I actually received the draft international standard from AMI yesterday. So that is making progress, and we are looking forward to providing the next round of comments. So one of the changes that I want to make sure I point out to folks is relates to the microbiological guidance in that document. In the prior version of ISO 17665-1, that guidance was very short. There was not a lot of information, and examples were either incomplete or non-existing. So that's one very notable change for the next revision of this document. There will be that microbiological validation guidance has been fleshed out, and examples have been provided that help to illustrate the science behind the microbiology for the validation process. That was fleshed out with input from pharmaceutical and medical device experts. And one of the things that was lacking in the prior version was examples and specifically calculation examples for F values. So those examples are much better than what was in there in the past. It covers, obviously, the overkill method, the partial cycle approach, the full cycle approach, cycle development. It covers the bioburden-based methods, specifically the combined biological indicator bioburden method and the strict bioburden method. And as I indicated, the examples were added for each method to better illustrate how to execute those validations. Some other documents that I want to mention. PDA Technical Report 1, that document's entitled Validation of Moist Heat Sterilization Process Cycle Design, Development, Qualification, and Ongoing Control. I think this document is a really good document because it breaks down general information relating to moist heat sterilization validation and moist heat sterilization in general. It gives good guidance on the microbiological aspects of that validation work. It also covers the physical science very well. It provides help for overkill and the bioburden-based approaches, and it contains more science than the ISO document. The ISO document primarily outlines requirements. The PDA documents provide, in my opinion, better scientific guidance. Another document for those that may be considering parametric release is PDA Technical Report 30, and guidance for parametric release for moist heat processes can be found in that document. Another one that I want to mention very quickly is the FDA guidance document for parametric release. So there are a lot of other documents. This list is not intended to be comprehensive, but these are good places to start if you have questions about moist heat sterilization validation. Another thing I wanted to do was to go over very quickly some FDA observations relating to moist heat sterilization. Interestingly enough, when reviewing a list of these observations, I was actually surprised at how simple they were. One of the trends that we saw was a total lack of validation data or missing validation data. So to me, that was surprising. There also were many observations relating to processing of tools, supplies, equipment that's used as part of the manufacturing process where proper validation had not been performed for those processes. So I think it's worth mentioning that when you are using a sterilization process in your manufacturing or as a terminal sterilization step, those processes need to be validated. Some of the things that you'll have to go through and are outlined in those documents that I mentioned and other applicable documents relate to microbiological characterization and performance qualification. You'll hear that often referred to as MPQ. Physical performance qualification, PPQ. For bioburden-based methods, which include the combined BI bioburden approach and the strict bioburden method, you need to have knowledge of your product bioburden. Depending on how you're validating and what you're doing, this will likely include enumeration, determination of organism types, specifically looking at spore formers. That may include ID work, and most importantly, resistance data, so that you can understand how resistant your bioburden is in a moist heat process. Another thing that you'll need to perform as part of that validation work is demonstration of biological indicator appropriateness. So you'll need to look at you'll need to compare the resistance of your biological indicator versus the resistance of your bioburden. And depending on the validation approach that you're taking, that may include comparison studies, fractional exposures, etc. Another trend that was observed when reviewing a list of deficiencies from the FDA has to do with appropriate qualification of equipment. And most of those observations actually relate back to a lack of equipment qualification. So, that qualification would include installation qualification, operational qualification, and performance qualification. It's important when you're performing qualification work and process validation that you look at your loading patterns. You may have a single load that you intend to sterilize routinely, and in a situation like that, the validation is quite straightforward. You would validate that loading pattern. But oftentimes you'll see that flexibility is required for the sterilization process. So you may need to look at minimum and maximum loading while you're going through your validation. You also need to look at proper chamber performance with each of those loads to ensure that you're able to achieve sterilization conditions. Another trend that was observed when reviewing FDA observations and warning letters was that there was a general lack of quality unit batch review and document review. That's an important part of product release, and that's something that you will need to build into the process. Additionally, if you have a depyrogenation process, and this likely will not apply to moist heat sterilization as much as it would a dry heat process, but I think it's worth noting that depyrogenation processes should also be validated to show that they perform as intended. And finally, one of the trends that was observed as well is a lack of proper equipment maintenance, establishing maintenance schedules, preventative maintenance, etc. So, I think one of the main takeaways is that when you have a sterilization process as part of your manufacturing, you need to make sure that those processes are validated appropriately. And Martel now will cover verification of sterility from terminally sterilized products. So Martel, go ahead.
Thanks, Jason. Hey, I wanted to mention one thing from when you and I were looking through those FDA deficiencies. It seemed that what we were seeing most often were the comments, just like you said, that a lot of those items were not addressed. it seemed that we did not see, and this is also my personal experience, we were not seeing situations where FDA was saying, you address this nicely in your files, but we disagree with one aspect of your rationale. It seemed that as long as a rationale was in place, as long as everything was addressed, generally that met their criteria, as opposed to being very specific with a specific item of a rationale. Was that your recollection as well?
Yeah, going through the list, it was interesting. As I mentioned, it was surprising to me because the trends that you and I observed, relate back to a complete lack of validation, a complete lack of justification. So, yeah, I would agree with you, Martel.
Yeah. As I work with customers, working through validations and rationales associated with those validations, sometimes we get really hung up on a really specific aspect of that validation. And I think this is a good reminder that really, as long as you document that you've addressed it and why you made the decision you made, that generally that's sufficient. And if they disagree, they might bring that up, and it'll be a more subtle comment from them, but will not result in the types of warning letters that we were seeing here.
Yeah, I also just want to mention that there are specific requirements when you validate a process, but I think that you're ahead of the curve if you actually have validation data as opposed to a lack of validation data. So
yeah, good. Okay, so for the second part here, we're just kind of targeting some specific things, sharing a little bit of some of the science or the big picture view here. But what we hope to get is a bunch of really good questions from those that are attending, that we can have a substantial discussion as part of the panel discussion or Q&A afterwards. So the few of the things that I'm going to touch on here, and this is something else that we see quite frequently. And the title there, you see verification of sterility in quotes, meaning so these are either components that a manufacturer might purchase to include in their kit or as part of their product. That's what I tend to see primarily with this pitfall here, is that their customer, their supplier, has already certified that the product is sterile. But to verify sterility, oftentimes the manufacturer that's purchased these pre-sterilized products will perform a test for sterility to verify that sterility. The problem with that, there are a couple big problems. The first is, for false positives, and working at a lab like Jason and I do, of course, we understand that we try really hard, of course, in our clean rooms to provide the best case scenario possible for performing a sterility test. But sometimes sterility testing is due to contamination during that test, or sterility test positives are due to contamination. But it's not the kind of contamination that you might think. Oftentimes, when you see positive sterility test results, the first idea that comes to mind is, well, did they drop it? Did they sneeze on it? Things like that. And that type of thing just generally does not happen. But what we do see is for products that have been sterilized and then they've been sent to a manufacturer to then use as part of their kit or just to sell if they have this third-party manufacturer manufacturing for them. Oftentimes that shipping process, sometimes the products are manually removed. Often they're removed from a shipper box. So the outside of these packages have been handled before they're sent to us for testing. And that means that usually the outside of the packages we receive for terminally sterilized products, the outside of that packaging is pretty highly contaminated with microorganisms. So of course, before we take it into the clean room, we're disinfecting the outside, we're being as careful as we can, but it's not a perfect process. And as we open these packages that have sat on someone's desk, for example, been put in a cardboard box and shipped to the lab for testing, as we open those in the clean room, it's very hard to first disinfect perfectly and then to open that without a single microorganism from the outside of that packaging to land on product or to contaminate the area. So that's one reason why, of course, we prefer to not perform those, because there is that chance for false positive. And then how do you deal with a positive? So proving that something's a false positive from a laboratory perspective is very difficult. Our experience with FDA is that having circumstantial evidence or evidence that information that might point to the fact that it's lab contamination is not enough. We have many instances where FDA comes to visit us, and when it comes to sterility testing, one of the first questions they ask is, we want you to share with us your failure investigations, especially all the ones where you said that it was or might have been laboratory contamination. And they dig into those really carefully. And so before we as a lab tell a customer, yes, this was laboratory contamination, FDA expects that the information, those data, they expect that to be almost concrete before we make that determination for our customer. Okay. So obviously, performing sterility testing on products or components that have already been sterilized is not best practice. There are better approaches that can and should be used. I'm going to talk about the science first of why that's not best practice. So the sterilization usually is to a sterility assurance level, or SAL, of 10 to the minus 6. This is a 1 in 1 million probability of a product being non-sterile. So you're trying to prove 1 in a million with a test of 10, 20, 40 samples. Let's say you test 20 samples, so 20 samples demonstrates an SAL of 10 to the minus 1.3 as far as sample size goes. So how do you bridge that gap between 10 to the minus 1.3, what you've actually tested, and 10 to the minus 6, which is what you're trying to verify? So how many samples would you have to test to actually prove, to verify 10 to the minus 6? Of course, at least 1 million samples, if not 3 million, if you want to do some statistics there. Some people say 3 million would be necessary. So graphically, here's what this looks like. Let's say this is either product bioburden or the bioburden of a biological indicator at 10 to the 6, so like 1 million CFU. And those of you that are familiar with microbiology and sterilization, you know, roughly we have a straight line. And so we're usually testing here at the beginning where we understand what the initial contamination or bioburden count is. And then we're testing somewhere here in the middle. And when we're actually testing for sterility. And the problem you see here, just to show it graphically, is you have all that dashed line below where testing is actually occurring, extrapolating to 10 to the minus 6, 1 in a million. So there's that big gap there between where you've tested and where you're trying to prove. And there's just no way to do that scientifically. So the better way to properly verify sterility, first is what the technical diligence of your supplier, or during your supplier audits. You need to have someone either there during the audit or that you have as a resource to prove during that audit, or to verify that at least these 4 items, that the initial validation is performed correctly, show that they're performing the routine sterilization process maintenance. So every form of sterilization has some type of routine maintenance that should be performed that involves testing. And that's always the case, whether it's a steam or gas option or radiation. There are routine things that should be done. And you can verify that your supplier is doing those things, that it's in their procedures. That's someone who's responsible for performing those. And then effective change management. That's sometimes the biggest problem, is that things start off really good, but then over time, things change, people change, and sometimes things did not get addressed properly regarding sterilization when they're making changes to some aspect of the product. And then some type of yearly check. So as the customer of your supplier, you can do some form of yearly check just to say, hey, you've been doing your requalifications, if it's radiation, if you've been doing your dose audits, but certainly not performing sterility testing. And I put a quote in here from ISO, the ISO standard on sterility. And this just explains the idea that this is just the last sentence there. So as such, performing a test of sterility on product that has been exposed to the complete sterilization process provides no scientifically usable data and is not recommended. And that's about as strongly worded as we could put in that ISO standard. But we wanted to make it very clear that there is no usefulness at all to performing sterility testing of product that's received the full sterilization process. So there's the last couple of slides here we have the Seymour Block had several versions, several editions of a book called Disinfection, Sterilization, and Preservation. The first 5 editions were edited by him. And for those of us in the sterilization microbiology world, and also in the preservation and the disinfection world, this book is kind of the Bible of those topics, and has been for decades. So the 6th edition that just came out a couple of years ago, in chapter 64, there was a new chapter added for bioburden assessment and tests for sterility. And I was fortunate enough to be asked to write that chapter, and I wrote that with a good friend of mine in the industry. And of course, this was reviewed, so it's not just me and Trevi Bryant, but this does reflect the thought process in the sterilization microbiology industry. That, this is a list of inappropriate uses of a product sterility test. And I won't go through the whole list there, but we see, as you look through that list, there are things that are very commonly where a sterility test is used commonly in those processes. But those, if it's a terminally sterilized product, these are all, in every situation, these are inappropriate uses of a sterility test. And then also in that same chapter, then there's a list of appropriate uses of a sterility test. And these are the things that some of these we've talked about. So you have mainly aseptic processing, where these are not terminally sterilized products. So for aseptic process validation and for batch release of a validated aseptic process, as well as during validation approaches for terminal sterilization processes. So when you're in the process of validating a process, this is a good time to test product for sterility, because those are not full processes. Those are fractional or partial processes. And then, of course, the last bullet, in some cases, you're using product sterility testing to verify a disinfection or a liquid chemical sterilization process. Okay. So one update here I just want to add. The last comment I have here regarding batch release of a previously validated aseptic process. We're in the process of revising the ISO standard to aseptic processing of healthcare products. It's 13408-1. And in that document, as far as I know, for the first time, at least in the ISO standards, for the first time, we're breaching this idea of having a parametric release of finished product for an aseptic process. So you have this is a situation where you have so many controls and monitors in place during that aseptic process that that the product sterility tests is completely useless. And there are a lot of us in the industry that wonder about the usefulness of the product sterility test as part of batch release of an aseptic process anyway. But in this case, we're actually providing a little bit of context of how someone in an aseptic process scenario might be able to rationalize the idea of a parametric release situation, so where there would be no product sterility test required post aseptic processing. Okay, so now the panel discussion. So Susan will be leading our panel discussion, and Jason and I will provide input along with her. So that's the end of what I was going to present. So Susan, I'll turn the time over to you.
I'm here. Thank you, Jason and Martel. That was really great, very informative. I actually can't wait to get to the October seminar to get more in-depth insight into some of these topics. So that's great. Let's just start right in. I kind of want to have a dialogue, and any practical experience that you can bring in will help. But I want to start off right away. There was a question: can sterilization parameters published by the sterilizer manufacturer be used in lieu of process validation? And if not, why not?
Yeah, that's something I would not recommend. This is Jason. That's something I would not recommend. That may help you to determine what parameters you want to validate depending on your product and what you need from your process. But it's important that you show that the equipment and the process produce sterile product using your product, your loading configuration, hooked up to your utilities, etc. So to summarize, I'm sorry, go ahead, Sue.
I was just, it's kind of like the process of compendial methods, USP methods are considered validated, but in order to use them in the lab, you have to verify that they're suitable for your lab. Is that kind of the same concept here?
Yes. Like I said, it allows you to ensure that you can achieve proper sterilization in your facility with the equipment that you control with your product, showing that those compendial parameters work for your process.
Martel, you have anything to add?
I don't. No, actually, that was covered very well.
Okay, great. Let's move on. Here's another question. We didn't really talk about particulate matter, but what role does particulate matter play in terminally sterilized products, and do I need to perform it? It's really critical, particulate matter, when you talk about aseptically built processes and products, but with terminally sterilized, how does that play into it?
Yeah, I'll take an initial shot at this one. It's interesting that sometimes we can get the idea that sterilization takes care of everything, but there are actually quite a few aspects of product where sterilization really is not beneficial. The main three that come to my mind are, from a biocompatibility standpoint, sterilization usually plays little to no role in the biocompatibility of a product. Bacterial endotoxin, in most cases, sterilization processes do little to nothing to reduce those. And then particulate matter is another one, where a product can be fully sterile, perfectly sterilized, but particulate matter, that's a different type of contamination as opposed to viable contamination. We would consider that non-viable contamination. And those certainly for some product types, those can be a big deal and should be addressed. And there are criteria in USP that covered, it gives at least some parameters around what you might do for some types of products. And unfortunately, there's not really good guidance for all types of products, even though particulates might be important. There are not really strict guidances or information on those requirements. So sometimes you're left to take what you can from USP or some other guidance and then find a way to apply it to your specific situation. So that's all I had. Sue, I don't know if you're muted or if you're.
Hi guys, I dropped off for some strange reason. I'm sorry for that technical difficulty.
Oh.
Where did we end up on particulate matter?
Well, yeah, welcome back. I just gave a great answer.
You did? Okay, good. Okay, so we're ready to move on to the next question?
Yes.
So what are your thoughts on best practices for microbial OOS investigation? And I just going to throw out here, are you aware of the new PDA technical report number 88 on microbial data deviation investigations in the pharmaceutical industry? It just came out this year, 2022.
Okay. Personally, I'm actually not familiar with that one. So I need to go and track that down. So thanks for pointing that out. So is there anything regarding that you'd like to bring up?
I think it's a really good overview with some in-depth details. I think that though this is worthy of a lot of discussion in some ways because they're one of the hardest, data deviate OS for the chemistry lab is a little bit easier. There's all kinds of structured ways to go about doing it, which apply in some ways to the micro lab, but because you're dealing with microbial contamination, it can be really difficult to find that root cause. So I just wanted to point out that there was this technical report, but in your experience, what's the best way to try to get to root cause, knowing that you're really probably going to come up with multiple potential root causes? And how do you eliminate that and say, no, the product wasn't contaminated, we can account for what happened?
Yeah, I have a few thoughts on that. Jason, should I go first and you can chime in?
Yeah, go ahead and jump in and I'll chime in after.
Yeah. I like that question because the smoking gun when it comes to OOS investigations is specific to microbiology. That smoking gun can be very elusive and often is very elusive. And the nice thing in some instances is that the regulatory bodies do not always expect you to find the smoking gun or the root cause for what caused it. But Sue, just like you mentioned, oftentimes finding the root cause can be very difficult or sometimes even impossible because you're often looking back at microbiology. And those of us that have done microbiology for a long time know that sometimes it is not very consistent. Sometimes something might be there because of something that was occurring at that time point that's not there now. So to do testing now, to try and understand what caused something a couple of weeks ago or a month ago, oftentimes is not beneficial or not fruitful. But what we do see, though, is that regulatory bodies do not expect you to have the smoking gun. They expect you to do a thorough investigation, and from the investigation determine these are the items that, with the best data that we have, these are the items that we think might have or could have contributed to this problem. So now we're going to implement corrective action, to address all of those items, and we're going to be very thorough on how we implement the corrective action, and then we can repeat that test with the corrective action implemented to verify if the corrective actions were effective. And we see that working well all the time. So very, very consistently. If you dig deep, find potential root causes, implement good corrective actions, then you can repeat testing and show that those were effective, and then move forward with whatever it was that you were doing. So those are the thoughts I wanted to share. Jason, anything to add?
I've been involved in a number of these investigations, and as you've indicated and as Sue indicated, I would say there's not one correct answer for where to start or how to proceed. They can be very difficult. They can require a lot of time to do a thorough investigation. What may be appropriate in one situation is not going to be appropriate in another. But when I've been involved and a thorough investigation has been conducted, I would say that in my opinion, those have always been good exercises. There's always been good outcomes because of that investigation, and processes have been improved because of those investigations. So that's really all I would add.
Well, they are very difficult, and I think they cause a lot of challenge to the industry to get those right. Let's move on to here's an interesting question that'll expand us beyond the pharma industry. So you talk about appropriate and inappropriate uses of sterility tests. Are those aligned across the pharma/medical device industry, including USP? And any kind of, I understand, rapid methodology is actually on the rise. Can you talk about how rapid methodology might help us in this situation?
Yeah. The first part of that question, the way I'd answer that is yes and no. So this list of items here, when I interact with other people in industry who are involved in writing standards and establishing requirements for pharma and device industry, conceptually, we are all aligned with these. If I talk specifically like package integrity and product shelf life, FDA has a document that's been out, it's like since 2008, I think, that you should be performing physical tests of the packaging to verify that the product is still sterile, rather than performing sterility testing. So this document's already been out for well over a decade. But we still see people performing sterility testing to try and improve shelf life. And there's so many potential pitfalls in the sterility test, whereas showing that the package still maintains integrity is by far a much better approach to take. And regarding rapid methods, let me finish up one aspect of that first question, the first part of that question. The difficult thing is that if you look carefully enough, you will find a couple of instances where official documents, either one or two from FDA, one from USP 71, where it implies that the product sterility test should be performed post sterilization. So even though in principle, as we sit around the table, we're talking about the importance of these things, we're all in alignment, yes. But occasionally you'll find something in a document that will lead you to believe that in some aspect, that in certain situations you should be performing a product sterility test of products that have been put through the full sterilization process. Now, Sue, can I have you repeat the part about rapid methods? I want to make sure I hit the right aspect of that.
Well, I've heard a lot about rapid methods coming into play here, that people are retooling their laboratories, micro laboratories, to incorporate rapid methods. And I'm just wondering, what experience we have in that, and is it a better indicator? How could it be a better indicator for measuring sterility than the current USP methodology?
Okay. Yeah, so the USP and the ISO methodology, they're both immersion of the filter or the product for 14 days. And yes, it's interesting scientifically, the rapid methods often are actually a better indicator of product sterility. And even though that's the case, we would still want to use those in these situations here as part of validation of either sterilization or an aseptic process, or batch release testing of aseptically produced products. But yeah, those it's funny because oftentimes you hear discussions about rapid methods and they say, well, they're not really well accepted yet, but yet when we have interactions with FDA, or when you look at specific guidance, you'll see that they actually really do want people to implement them because of the in most cases the heightened sensitivity of the rapid methods and the greater speed at which you can obtain data to be able to make informed decisions earlier in a process rather than later. So from a regulatory perspective, they're very much on board, and it's still a little bit difficult sometimes to understand how you need to implement that, but they are well accepted and should be considered going forward. Does that answer that, you think, Sue?
Yeah, I think it does.
Jason, do you have anything to add to either of those two?
No, I think you covered that very well, Martel.
Okay, so I was really interested regarding the concept of ISO 13408-1 and parametric release of aseptically filled products. So will that encompass manually filled aseptic products also? And two, what are some of the parameters you might be looking at that would be key in being able to achieve that? That's very ambitious, and I applaud ISO for taking it on.
Yeah, it's funny. I was in let's see, it was an AMI meeting, so it was not an international meeting, an AMI meeting with a specific FDA reviewer. I won't mention him by name, but this FDA representative was in this meeting. We're talking, and this was 15 years ago. And back then, even he brought up as an FDA reviewer, he said, and heaven forbid that we actually start implementing really good science and do parametric release of aseptically processed products. So that concept, even in the mind of some FDA people, has been present for a long time. But as the consulting side of what I do through Nelson Labs, I've been involved with many situations, different types of aseptic processes. And I have to admit, I've been on site to a couple where I look at that process and I say, well, this might meet criteria for aseptic process for like performing a bioburden test or something like that. But for a product that is supposed to be sterile afterwards, this is completely not even close to an aseptic process. And the reason why, and so you hit that right on the head, the reason why it was very manual in nature. And the more manual a process, an aseptic process is, the less and less likely that you'd be able to implement a parametric release approach to products that go through that process. The example that we give in the ISO standard that will, and it's not completed yet, but there, it just went through another review, another round of comments internationally, and so far there have been no strong concerns with this concept. That in cases where it's all automated, you're doing, wherever there's a potential for contamination, it's being done in an isolator, and in most cases they're even closed systems in an isolator. So where would you have the opportunity for contamination, where you've got filtration occurring during the process, all the other components have been sterilized prior to incorporation into the process, and it's a very clean process, very well thought out. A lot of controls are in place. A lot of monitors are being performed during that process with regards to environment and those few people that do have some interaction with the product during the process. Those are the types of aseptic processes where the problem is the product sterility test is being performed in as good, or in some cases in not as good a situation, just because you cannot always recreate a closed system in an isolator for some types of products. So what benefit would there be for a product in that scenario? What benefit would there be to a product sterility test? There is no benefit. So that's the kind of scenario we're talking about there. Did I cover that, Sue?
Yeah, I think that's great. So one final question, I think, before we leave. What role, media fills are really important in aseptic processing and any kind of manual aseptic processing. What role do media fills have in a terminally sterilized process, if any? Okay,
so I'm going to change the term first from media fill to process simulation. The reason why is
media fill, I like that.
Yeah, well, so media fill has been the traditional term, and it still is fully applicable to many types of aseptically produced products, because you're filling the vial with media instead of product. But in the last quite a few years, we're seeing a lot of combo products, combination products that involve a biologic or a drug, and these products are sometimes they're proven to not. You just cannot put those through an aseptic, you cannot put those through any type of sterilization process that does not negatively impact the effect of the pharmaceutical or of the biologic component of that combination product. And so they're forced into aseptic processing. And I should add, in the industry, we're trying to make it clear that it's not always terminal sterilization to 10 to the minus 6 or aseptic processing. That there are documents in the industry to support that. Rather than going from 10 to the minus 6, we can't do 10 to the minus 6, let's just go to aseptic. An alternate SAL, for example, 10 to the minus 5, 4, or even 10 to the minus 3, still provides assurance of sterility that is actually more easily demonstratable than an aseptic process. So you could rationalize use of 10 to the minus 3, for example, a terminal sterilization process, and still put sterile on the label rather than going with aseptic. But that's the reason that that I prefer to use the term nowadays, prefer to use the term process simulation, because those types of products that have biologics or drugs on them, you can't replace product with media. In this case, you're performing a product sterility test as part of that aseptic process. So now, how does that play a role in terminal sterilization? It actually does not. It plays no role. But you could implement product sterility testing. In terminal sterilization, you will be testing product for sterility, but at shorter sterilization cycles, a shorter process, a fractional process. But they're really the purpose for media fill or process simulation is to show that once a product goes through the entire process, the product is sterile. And we've just talked about when you would not be, that there are inappropriate uses of a product sterility test, and that those are the inappropriate uses of, if a product has been terminally sterilized, no benefit to performing product sterility. So really the terms media fill or process simulation are reserved only for situations that are involved with aseptic processing rather than terminal sterilization. So I don't know if that makes sense. anything to add to that?
I do have something to add to that. There are times when we validate a terminally sterilized or terminal sterilization process that we might use one liquid as a replacement for another during that validation. If we do that, we need to make sure that we demonstrate that both of those liquids perform the same when they're heated. You may have a liquid where it delivers less lethality when heated versus another liquid. So doing those comparison studies is important.
Great. I think that's about all we have time for. So I want to thank you guys for an excellent presentation and an excellent discussion on the questions and answers. Tim, I'll turn it over to you.
All right, great. Thank you so much, Sue. Really appreciate you leading that discussion there. Covered a lot of ground. And thank you, Jason and Martel, for the slide deck presentation. You guys did a great job. Just wanted, before we wrap things up, I want to say thanks again to today's sponsor, Nelson Labs. You want to be sure to visit them at nelsonlabs.com. And I'm going to direct you to the second slide up here. I believe I want to, yep, there we are. If you are interested, I'd like to direct you to the seminar section of nelsonlabs.com where you can register for this session that they'll be hosting here. Just one last thing before we go. Today's session will be archived for 1 year on our website, contractpharma.com, and you'll be receiving an email from us shortly when the session's available for on-demand viewing. And if you are interested in downloading a PDF of today's PowerPoint presentation, that's available for you in the Resources tab on your screen there. So thank you very much, everyone in the audience, for your attendance and participation today. We very much appreciate you joining us. Have a great rest of your day. Take care.